Hi, everybody. Welcome back. We are entering to chapter 2 in our psychology quest for knowledge. Chapter 2 is my personal favorite chapter, neuroscience. I don't know if you guys remember or if you even looked at my background, but I actually have my PhD in neuroscience. So this is my specialty. I love it. It's my favorite chapter. I think it's the most interesting. And in this chapter, we're going to kind of learn all about the most important organ in the body, which is the brain, which is responsible for everything that happens in our body, our behavior, our thinking, our feeling. A lot of people think that the heart is really important. I'm sure like heart surgeons think that. It's not. It's basically just a giant muscle that pumps blood. The brain is responsible for all of our functioning, our thinking, our feeling, our automated systems, right? All of our hormone systems, our breathing. The brain actually controls the heart. So just saying, the brain is the most important. But anyways, this is kind of a cool chapter to me. So let's go ahead and get started. In this chapter, of course, you'll have your chapter objectives. We're going to look at structure and function of neurons, axons, dendrites. You'll be able to identify all of those parts of a neuron. I'll explain to you what an action potential is and how we communicate in our brain, how the cells of our brain communicate with one another. We'll look at the processes of both excitation and inhibition at the point of the synapse. So the synapse is where two neurons meet. They don't actually touch, it's a little gap, but we'll talk more about that. You'll be able to list all of the major neurotransmitters and neuromodulators. Those are the chemical messengers in our brain, and describe their function. You'll also be able to describe the major parts of the nervous systems and how information is processed within. You will be able to locate the different areas of the brain: hindbrain, midbrain, forebrain, and explain all of their parts, all the neuroanatomy and the functioning of those systems. You'll be able to describe what happens, what changes as our brains age, and what is known about protecting the brain as we age. We'll look at some brain imaging techniques and look at the ways of how we study the brain with advantages and limitations. There's not always a perfect way to study that. We saw that with the research methods. There's a lot of different ways to ask questions. Finally, we'll end up talking about the endocrine system. So that's your hormone system and how those glands work. So this is section 2.1 in chapter 2. So neuroscience itself is just the study of how the brain and the nervous systems impact not only our mental processes, but also our behaviors. So the brain is made up of specialized cells called neurons. So these are just very specialized in their function, particularly in their shape. Their shape impacts their function. So they transmit information throughout the nervous system. So that's from our brain down to our spinal cord, throughout our whole body. We also have glial cells. So there's different types of glial cells. Glial cells are kind of the support cells in the brain. You've probably heard of like gray matter and white matter when we're talking about the brain. So the gray matter are the neurons themselves. The white matter is the glial cells. And then myelin is actually this kind of fatty tissue substance that's wrapped around the axon of the neuron. And that's really important for protecting that axon. So here's kind of a cartoonized picture of what a neuron looks like. There are different areas which have been named. So you have this top portion, which kind of looks like the medusa, that's the head. That is the round part in the middle is the cell body. Inside that cell body, you have the nucleus, which contains all the genetic material. So the blueprint, if you will, for our bodies and our systems. The dendrites are the things that are coming off, and those are actually going to receive information from neighboring cells. The long kind of looks like hot dog wrap, this is the axon. Okay, the axon is actually coated in a myelin sheath. And then you get to the what we call, there's a lot of different names for this bottom part. That's where the message is sent to the next neuron. So these are called, they can be called axon bulbs, terminal boutons, terminal buttons, terminal bulbs, but you get the idea. That's where the cell ends. And then you'll see the synapse right here. So a terminal button actually will synapse onto a dendrite of the next cell in the chain. So it's kind of like playing telephone. You pass the message down. But just remember, there's a little gap there. So a synapse that we used to believe, because we didn't have the equipment that was strong enough, like the strong enough microscope. So when people first started looking at neurons in the brain, they thought that the cells were touching. But as we developed more powerful microscopes and kind of zeroed in and focused down, it's actually a little space. All right, so we'll keep on going. So neurons communicate in electrochemical communication. So electrochemical, so there's electricity. They didn't know your brain had electricity, and there are chemicals. So the chemical part is actually the neurotransmitters. Neurotransmitters are released at those terminal boutons, and they float across that tiny little gap, and they reach the other cell. So you have a synapse, you have a presynaptic neuron, and a postsynaptic neuron. The presynaptic neuron is sending the messages, the neurotransmitters. The postsynaptic neuron is receiving the messages, okay? So again, we have that synaptic gap in between. Presynaptic, the one sending messages, postsynaptic neuron receiving. This is a little bit closer look at that synapse. So here you can see this terminal, the axon bulb or terminal bouton, and it is filled with synaptic vesicles. So these are kind of like tiny little pockets that hold a little vesicle that holds the chemical messengers, the neurotransmitters. So when it gets to the edge, it receives the nerve impulse. So that's the electrical part. And that tells the system to push those bulbs to the edge, where they kind of like basically like are emptying out their pockets into this gap. And so then these bulbs reach the edge, they open up, there's a mechanism to open and release those chemicals into the synapse, and then they can be received, they connect to receptors on this postsynaptic neuron. Okay, so how do neurons have electricity? Well, there are certain chemicals in our bodies, and they're made up of ions. You've probably heard of sodium, and you've probably heard of potassium. So these are ions that play a very important role in sending signals. This I do have some extra videos in there specifically about the I think I called them bonus videos that are in regard to the membrane potential, the resting membrane potential, talking about the action potential. So this topic is probably the most difficult for people to wrap their head around. So don't feel bad if you're like, I have no idea what's going on. This is electrophysiology. I am freaking out. Don't be scared, okay? It just takes a minute to digest this information. So remember, you do have those bonus videos. Anyhow, the membrane of the axons sits at a resting state of -70 millivolts, okay? So it's sitting at this resting, and a potential means the difference in charge across the membrane. So outside the membrane and inside the membrane. So it's sitting at a -70 inside the membrane, okay? And this is just across, like, think of the membrane as like the outer skin of the neuron. So it's sitting there at -70, okay? That's the resting potential. When the potential changes, so the axon has all of these little channels and they can open. And so when those channels open, okay, when those receptors indicate that the channel should open, you have a rush of these positive ions, sodium and potassium, rushing into the cell. And that causes the change. And that is what the action potential is. So that's how the action potential fires. Okay, so it goes very quickly from -70, rush of positive ions inside, all the way to +55, and that neuron says, boom, I'm sending a signal, okay? But it has to reach the threshold. You can't just go from -70 to -20. That's not enough. It has to be this large rush, and action potentials are considered all or none. So they either fire or they don't. They don't fire a little or halfway fire, or one fire is bigger than the other. It's all or none. It's either open, we flood and open everything, or we don't open it all and the signal is not sent. At the end of this flush, there's a thing called a refractory period. So the cell has to reset. The neuron has to reset back to -70. And there's some mechanisms by which that works. The cell has to reset. So you have this very brief, what's called a refractory period. You cannot fire another action potential while the cell is resetting. Okay. I know that's a lot. Bear with me. We're going to keep going. So here is an example of our axon. So think of this long tube, that's an axon. So you have sodium, mostly sodium. So sodium, some of you may know that sodium chloride, NaCl, is salt. So salt is very important to our body's functioning. If we had no salt in our body, our brains would not work. So too much salt is bad, but not enough salt, and you have some dysfunction in the system. Anyway, the sodium is on the outside of the cell and it's in a higher gradient, okay? So you have more sodium on the outside. Think about if you had too much sodium inside the cell, what would happen? So think about when you eat a lot of salt and what happens, you're gonna get bloated, right? So the same thing would happen to the neuron. So we keep the salt high, the sodium high on the outside. And there are all these channels, right? So we have this negative resting potential, positive on the outside, negative on the inside. As we said, it's -70 millivolts, okay? So what happens is the cell will receive a stimulus, okay? And this is probably from neurotransmitters that are landing on the postsynaptic cell saying, hey, open the sodium channels. That's the stimulus, open the sodium channels. We need to send a message. So these sodium channels open and they rush in, okay? They rush in. And so that sends the signal for the channels that along the edge or along the axon to open. So it's kind of like a domino effect. So these first ones open and then it sends a signal, okay, these open, these open, these open. So now you have this rush of sodium inside the cell and it propagates or travels along the length of that axon, okay? So we're opening channels, send the message, push all the way down. Remember, we're getting the signal from the dendrites at the top of the head, sending it down the axon to the axon bulbs at the end, the terminal boutons. I just like saying bouton, it's like fancy. This is a diagram of the action potential itself. So whenever you're looking at a graph, you always want to kind of identify, okay, what's on the x-axis and what's on the y-axis. Okay, so on our x-axis, we have time. So this is just time proceeding. On the y-axis, we have voltage. So here we have millivolts. Okay, so of course, -70, that's where our axon, that's where our resting potential lies. We have marked here -55. So that's going to be your threshold. Once you reach threshold, all the sodium channels open. The millivolts pop up to a +40. So this was a rush of all the sodium channels. Then you have potassium, which is inside the cell. Then you have those channels start to open. So potassium is different than sodium. Sodium is big on the outside. There's a lot of sodium on the outside. Potassium has higher concentrations on the inside. So when those channels open, potassium starts to leave the cell. Now, both of these ions are positive. So when sodium enters, we have a positive impact. When potassium, also a positive, starts to leave, you have a negative. It starts to go back negative because you're losing those positive ions because it's following along its concentration gradient. The sodium channels close here at this peak because they say, we've had enough, stop, we're good, they close. But then, then when the potassium ions, the potassium channels open, the potassium starts to leave and we fall down this negative. So we go back, we go a little bit too far. I always like to think of sodium like the mom and potassium's like the dad. Like the mom sees the signal like way earlier and then she's like, I've had it, I can't deal with this. And then the dad kind of comes down and he's like, what happened? What's going on? Hey, everybody calm down. So mom, sodium, dad, potassium, try to be the good guy, but maybe he just lets the channels stay open a little too long. We go a little too negative. So then we have to work back up to our kind of resting membrane potential at -70. Okay, so we've just talked about the action potential, which is going to travel down the length of this axon. Once it gets to the bottom, that is the signal. So all of those positive, negative, positive channels opening and sodium rushing in, that sends the signal to the cell to push the vesicles to the edge and release neurotransmitter into the synapse. The neurotransmitter then floats across and attaches to its specific receptor. So every neurotransmitter, the shape of it has a specific receptor, and they just connect in perfect, like a lock and key, okay? So based on the shape, it's gonna connect to that receptor, and it's gonna cause the next cell, the postsynaptic cell, to go through this process. Open sodium channels, push the signal, and go to the next. So that's how these neurons communicate in sequence. When we have synaptic transmission, that's what it's called, kind of the transmission of the signal from cell to cell, there's excitation and there's inhibition, okay? So we need both in the system. So excitation means that the postsynaptic cell, it becomes more positive inside and is going to send the signal. Inhibition is going to cause the cell to not fire. So you have kind of, think of it as like a cross, right? You have to have, if you have a green light here, you have to have a red light so that people don't crash, right? So if we're a green light, like, nope, we're just traveling south, we're going to turn on the red light for east and west because we don't want to send mixed signals, okay? So we need inhibition to help moderate the system. Any neuron can receive both excitatory and inhibitory signals. It would just be which one is stronger, determining what that cell will then do. If it's more excitation, the cell will fire an action potential. If it's more inhibition, the cell will not fire. So after this, remember, we've dumped out our neurotransmitters into the synapse, right? And so we have to clean them up. So we a lot of times we recycle those neurotransmitters. So they were just chemicals out in the synapse, and it's like, okay, well, we need to kind of clean those up. So we have a system called reuptake, where those neurotransmitters that were floating out in the synapse, they are actually taken back into the presynaptic cell. So it's basically like, okay, you dump all the marbles out of your pockets and you let the marbles do what they're going to do, and then you go around and collect them and bring them back into the cell so that they're ready for the next time the cell has to fire. There are also some times where neurotransmitters are just broken down because they float away, and so they can't be taken back up. So there are different proteins and chemicals that will just break down those and break them into their individual parts, and then they will be recycled through our body. Okay, I know this was a lot. So here's your first written reflection for chapter 2. Talking about neurons and neural circuits. We talked a little bit about why both the excitatory and inhibitory inputs. So first, I want to answer these 3 questions. Why is inhibition necessary in the brain? What would it be like if there was only excitation? So think about that. And then how do excitation and inhibitory inputs work together? Remember, there are some extra videos on the action potential and the anatomy of the brain, the bonus videos. So if you were kind of lost in that part, please take a look at those. They're very helpful. I created them. They're like little cartoon. They're cute. So make sure you're jotting down your answers for your written reflection, and I'll see you for the next video.